Hi, everybody. My name is Jim Shaw. I'm a postdoc at Dana-Farber Cancer Institute in Harvard Medical School, and I'll be talking to you today about how new computational methods can unlock high-resolution nanopore microbiome-based analyses. So today I'll be talking about microbiome analysis, such as metabarcoding and metagenomics. So the picture looks like this. We have a microbiome sample. We extract some DNA, and then after we extract this DNA, we can subject it to various types of analyses. One type of analysis is to do shotgun DNA sequencing, and this is called metagenomics, where we have a whole genome untargeted sequencing of all the genomic content. And on the other hand, we could do something like amplify conserved marker genes, for example, the 16S rRNA gene, and then we can sequence those amplicons, and this is called metabarcoding. So today I'll be talking about both types of analyses and why we want to use long reads instead of short reads. So for example, for metagenomics, one thing we want to do is assemble the reads into genomes, recover the genomes of interest. And historically, for short read assembly, it's well known that we end up getting these bins of these contigs that can be quite fragmented. These are also called mags. For long read assembly, we could potentially get more complete, sometimes even circular genomes. On the metabarcoding side, if we're interested in targeting a specific gene like the 16S gene, short reads can only sequence a fragment of this gene, whereas long reads will allow us to sequence amplicons for the entire gene. And this is important if we are interested in distinguishing 16S copies from similar strains or species, because there might be only a little bit of genetic variation. And if our short reads capture only a small fragment, we're not able to see this variation, right? So long reads can give us a lot of potential in resolving things at higher resolution. Now, in the past, sequencing errors have been an issue for long read analysis. For example, on the metabarcoding side, you can imagine if only two substitutions differentiate these two strains, if I have a lot of errors, it might be challenging to kind of assign these reads to the right strain. On the metagenomic side, errors can kind of propagate down to the assembly level, and errors can also fragment the assemblies. But with modern nanopore reads, right, the R10.4, the latest chemistries, the latest base calling, these reads are actually very accurate now, over 99% accurate on average. And this gives us some interesting opportunities to work with. So today I'll be talking about taking advantage of modern nanopore reads by choosing the right computational tools. So we've built some new interesting computational tools. For example, I'll be talking about how to achieve strain-level nanopore metagenome assembly today. And I'll also be talking about, on the amplicon sequencing side, how to get these ASVs from nanopore amplicon sequencing. And the theme, again, is to take advantage of these modern reads by using modern computational tools. And I'll show that it's necessary to not just think about the technologies, but also the computational side. Great. So the first thing I'll talk about today is Myloasm. This is our new metagenomic noisy long read assembler. And if you recall, assembly is the process of turning these shotgun reads into hopefully circular complete genomes and or these mags. And the motivation of Myloasm is that there's a lot of sequence homology between genomes in a microbiome. Right? You can imagine on the mobile genetic element side that there can be prophages, transposons, all sorts of things shared between distinct species that can be very, very similar due to horizontal gene transfer. And you can also imagine that there's coexisting strains of the same species, right? 95 to 99, very similar DNA between coexisting strains. And for assembly, this is a particularly challenging problem, right? But with modern nanopore reads, again, we can see, is it possible to resolve this highly similar genomic content? And I'll talk a little bit about the bioinformatics for the next 2 minutes before jumping onto the results. But the principal problem is, how do we know if two reads come from the same strain? Imagine I have two E. coli strains hanging out in my microbiome, and they're very similar, and I just get two sequencing reads. Do they come from the same strain, or do they come from similar strains? Well, because even with modern nanopore reads, there's still some sort of sequencing error. It can be hard to disentangle, do these reads come from the same strain or different strain, right? On average, if reads come from the same strain, they'll be more similar. If they're on different strains, then they'll be more dissimilar, but still the distributions of kind of this dissimilarity, they overlap, right? So the idea here from the bioinformatics side is to use sequencing redundancy to find true genomic polymorphism. And what I mean by this is, let's say I have these strains, they're sequenced multiple times to a certain depth of coverage. There will be random sequencing errors along these reads, but there will also be true genomic differences in these colored boxes. And the idea is here is how do I capture these true genomic differences? And one thing I can do is I can look at k-length substrings, right? Sequences of length k of nucleotides. These are called k-mers. And I want to capture nucleotides where they're almost the same, but there is this substitution in the middle. And what this tells me is that if I see this occur very often, it's likely that there's a single nucleotide polymorphism within my sample. So that's why we call these substrings SNPmers. We can capture these SNPs, single nucleotide polymorphisms, in this k-mer context. And by doing this, I can analyze just these substitutions very closely. I can discard the other information and just focus on the polymorphic part, right? It's a form of error correction or signal refinement. And with these SNPmers, I can disentangle overlaps. If you recall, with raw overlap similarity, it can be quite ambiguous, right? I can't tell if two reads come from the same strains. But if I just focus on that polymorphic region, I'm able to disentangle these distributions. In other words, I can tell if two reads come from the same strain because they share all the SNPmers. And so this is just, I'm kind of flashing a slide here to show some of the methodological details that you can see in the paper. On the left-hand side, we do some more complicated things. We've used sequencing depth and kind of have this assembly graph that we traverse in a probabilistic fashion. We use some more mathematical tools to deal with this. But focusing on the results here, I just wanted to emphasize, my initial point, which is that previous methods do in fact struggle for coexisting strains within the community. So this is a Nanopore mock community that I'm showing you here with various strains sequenced to depth, as you can see on the left-hand side. And I just want to focus on these two species. So in my community, there was 2 Staph aureus strains and 2 Listeria monocytogene strains. And what you can see is that with previous methods that are state-of-the-art, like MetaFly, they actually miss out on the lower abundance strain, even though it's sequenced 50x times, right? And even when it's able to recover it, you can see over here, the Q-score for MetaFly is a little bit lower. Now, Q-score is actually on a log scale, so it's about 200 times worse than Myloasm. Okay. And so what you can see is that previous methods struggled for this high resolution task, and it's not their problem, right? It's not the algorithm is bad. It's just that the algorithm was designed for older sequencing data and wasn't adapted to deal with this high quality data that we can take advantage of with Myloasm. And one particular story I like to highlight here is Nanopore versus PacBio HiFi. So in this wonderful study by Minnick et al. That came out last year, they sequenced 47 Malawian gut metagenomes with both technologies. And they actually found that with PacBio HiFi, they got 2.3 times more circular genomes than Nanopore. But this study came out before Myloasm was published. So we want to ask, with Myloasm, does this picture change? And in fact, it does change. We found that with Myloasm, on their exact same data, Nanopore can actually surpass HiFi for circular complete contigs or genomes in the gut. So on the y-axis, I'm showing you the number of circular high-quality contigs or genomes recovered as a function of sequencing depth. There's 2 samples. There's D5 on the left-hand side here and H6 on the right-hand side here, and HiFi and ONT for both samples. And what you can see is that compared to the previous methods on HiFi, Myloasm is doing a little bit better or possibly similarly. But on the ONT data, we're actually recovering way more genomes. And on these dashed lines, I'm showing you that at the highest sequencing depth, there's more circular contigs with ONT than HiFi. And overall, we get 3 to 5 times more circular contigs than previous tools for Nanopore R10.4, which, as you can see by these frames here. Okay, so the sequencing technology is fantastic, but you also have to choose the right tools that can take advantage of these new technologies. All right, so one cool application that long reads in Myloasm can allow you to do is to resolve mobile genetic elements and antibiotic resistance. This was done with my fellow postdoc, Max Marin. So what I'm showing you on the left-hand side here is this phylogenetic tree of the Bacteroidota phylum. And I'm actually showing you 19 circular Bacteroidota genomes and 4 non-circular genomes assembled from a single sample. And after doing some antibiotic resistance analysis, we found this ERMF or erythromycin resistance gene being spread throughout these clades. And upon closer analysis, what we found was that there were 2 classes of mobile genetic elements spreading this ERMF gene. But because we had long reads and this was a very difficult region to assemble, with MyloASM, we could actually assemble the genetic context of these MGEs and found that there's two classes of MGEs spreading this resistance, right? So we found with previous tools, there were difficulties resolving this complicated locus. So this was very cool. And the last project I want to talk about is not actually our project. It's a recent paper released by Louis and Nielsen. And what they did was they applied MyloASM to a terabase scale metagenome assembly. They applied MyloASM to 720 gigabases of nanopore sequencing, and they actually assembled a ton of circular high quality stuff. And they found that we assembled way more circular and high quality contigs with MyloASM compared to the previous method. So in conclusion, on the metagenome side of my presentation, I want to show that with MyloASM, with new tools and modern long reads, you can get a lot of interesting things, right? Newer types of analyses, mobile genetic elements, antibiotic resistance, and it's starting to become competitive with HiFi. So I think this is a great indication of kind of where the field is going, and this is a very rewarding project. Our paper is available, and also the software is open source and available, as you can see here. So now I want to finish the presentation by jumping onto the amplicon sequencing side. So if you recall, let's say I want to sequence a specific marker gene like the 16S. I get these long read amplicon sequences, but again, I'll have sequencing error. Now, the goal on the amplicon side is I want to resolve these amplicons. I want to remove these errors and generate what are called amplicon sequence variants, or ASVs. And once I get these ASVs that are free of the sequencing error, I can profile the community at high resolution, hopefully with these long, nice 16S ASVs. The motivation for this is that previous studies have shown that you get much better taxonomic resolution if you use the full-length 16S gene compared to just short reads. For example, this is a very famous figure from this paper by Johnson et al., where they show that across the phylogenetic kind of tree of life here, red is the percent that's unclassified as a function of the specific region of the 16S gene. So what's important to us is that for the V1, V9, this is the entire gene, almost all of this tree is white, showing that you can resolve things at high taxonomic resolution. The problem, though, is that previously the Nanopore ASV algorithms just didn't exist. These denoising algorithms, such as DADA and unoise, they work great on low error, short length amplicon sequences because they rely on the existence of perfect amplicon sequences without any errors. But as your reads get longer and longer, you're more likely to get some errors within these amplicon sequences, which makes those previous methods not work as well. So what we did was we created a new method. It's called Savant, right? ASVs from ONT. And the goal is to resolve these ASVs, and we use SNPmers again. So this is almost exactly the same picture as I showed before, capturing those polymorphisms using those K-mer contexts. Now, I won't go into the method at too high level of a detail, but the principle is pretty simple. You use those SNPmers like before. You find clusters of similar reads using those SNPmers at high resolution. You get a consensus with these clusters of reads, and then those consensuses, we filter them out based on chimeras and poor quality consensuses to get the final ASVs. Right? So it's a pretty simple approach, and what we found was that it was very effective for ONT data. So on a mock nanopore community, what we found was that on this left-hand side here, it required 16 times lower sequencing depth in order to generate ASVs compared to unoise, which was the previous denoising algorithm. On the x-axis, this is the depth of the ASV you need in order to get to recover the ASV. And it turns out that our ASVs are still very precise, and we're much more sensitive, but at the same time, the ASVs are high quality. They look like the reference sequences. So this is on mock data, and when we extended this to real data, we confirmed that Savant detects much more diversity at lower sequencing depths. So for 4 types of samples here, anaerobic digesters, wastewater, fecal samples, and soil, on the x-axis is the number of reads that we downsample or rarefacted. And what you can see on the top is the Shannon diversity of these communities at the ASV level. In other words, when you have low sequencing depth, Savant's able to capture much more of the Shannon diversity than previous methods and gets you a much higher number of ASVs. And on the profiling side, so that was just ASV generation. Now I'm interested in capturing species based on the ASVs. What we found was that these ASV approaches can lead to fewer false positive than previous read mapping approaches. So the previous state-of-the-art, such as EMU, relies on mapping reads to the reference. But for these profiling methods, you get this long tail of low abundance false positives that we show over here, right? And with ASV methods, you're able to avoid some of this noise by denoising the amplicons beforehand. And finally, Savant's easy to use and available on GitHub. You can download, classify your against databases, and also generate ASVs. So I'll wrap up by saying that with modern nanopore sequencing, we can get these ASVs at low depth, and we can really enable this ASV-typed amplicon analyses. And this is a collaboration between our fellow colleagues at Aalborg University. So to wrap up today's talk was about taking advantage of modern nanopore sequencing by choosing the right computational tools. I showed how metagenomes and metabarcoding, you can kind of get better results by choosing modern computational tools and taking advantage of modern long reads. And just to recap, there's that theme I talked about today. So finally, I'll end this presentation with some thanks. Thank you to our lab, Hung Lee, who's my supervisor at Dana-Farber and Harvard, Max Marin, who I also worked with on the metagenome project, and our wonderful collaborators in Denmark at Aalborg University, who have helped us a lot on the application and data generation side. Thank you very much.